Hello, students. This is Dr. Faizan Mirza. In this video, I'll be covering with you how respirometer can be used in investigating the rate of respiration of an organism. So this question came in October 2013, video 52. So the question gave you a diagram of a respirometer, a simplistic respirometer, something that you already discussed in your content. So a student used a respirometer shown in figure 1.1 to compare the rate of respiration of three organisms: the germinating seed, the insect larvae, and single cell green algae living in water. So this is you can see the organism being placed here. So there's a carbon dioxide absorbent there. These tablets are there and you can weigh them and place them. So you know how much carbon dioxide absorbent you are placing there. There's a water bath to ensure that it will make sure the temperature of the water stays constant. Plus the graduated tube, you can see it's filled with water. You can just see the meniscus here. The meniscus actually tells you that this water is already there. So as the organism will keep on respiring, it will keep on using up the oxygen here and the CO2 will be absorbed here. So the oxygen from the tube will be used up and the meniscus will start to fill up with water. So you can see the meniscus will start to move into the tube. So you can see the water moving into the tube and you can note down the time taken from here to here or the distance covered in known times, either one which can do, which you normally do for a respirometer. So after putting the germinating seeds into the air-filled jar in the container attaching the graduate tube, the respirometer was lowered into a water bath. The seeds respired using oxygen and water moved into the graduated tube. The procedure was repeated for other two organisms, suggests the hypothesis about the respiration of different organisms that the student could use could test using this apparatus. So they're asking for the hypothesis, not the null hypothesis. We will say there is an association and there is a significant association. So, what is the hypothesis? The rate of respiration is significantly affected by the type of organism used. So that will be if they had asked for a null hypothesis, our statement would have been the rate of respiration is significantly not affected by the type of organism used. So identify the dependent variable. Independent variable here the type of organism, which is either the generating seed or the insect larvae or the single cell organism. Again, the independent variable is the cause. And what is the effect? What effect do we see here? The effect we see in this speedometer is a dependent variable. Is the distance moved by the air bubble? In this case, it's not the air bubble, it's the water meniscus. Normally in respirometer, we have the air bubble that we use. But here, since we are using the water meniscus to move, so we know how the distance moved by the water meniscus along the capillary tube in a known time, it will be our dependent variable. Describe a method the student could use in respirometer figure 1.1 that the student could use to compare the rates of respiration. It should be descriptive enough. So yeah, you would need to make an 8-mark answer here, including all the variable procedure, safety, and reliability. So you can say that you can mention how the organism can be weighed. So if you take the insect larvae first, weight and place it in a dish inside it, and lower this apparatus into the water bath. The carbon dioxide absorbent should be fresh and weighed, and its mass should be known. The mass of organism place is known as well, and you are using insect larvae, for example, initially. So, and you are placing it in a dish, and you will make this airtight. You can use Vaseline for this, or any kind of substance which makes sure that there is no air moving into it. And the graduated tube is carefully and is actually in lowered back. The initial meniscus is noted for the water, and then what you do, you just allow the organism to acclimatize to the new environment. You leave it for 2 minutes so it adjusts there. After 2 minutes, you start to note down the distance traveled by this water meniscus in known time. For example, you just leave it for 10 minutes, and after 10 minutes, you note down how much distance this water meniscus covered. So this will give you one reading. Now you should further mention for the reliability, repeat it 2 more times. That means a total of thrice using the same organism type but a separate organism, a different organism of the same type. You need to take a fresh organism every time. You will not keep repeating with the same one. So you take three separate readings. The carbon dioxide absorbent need to be placed fresh every time, and it must be of the same mass that you had taken initially. The mass of the organism should also stay same when you're replacing it. Again, you will repeat the whole procedure. Don't give the whole description all over again. Just give the brief overview that I've just discussed. And once this is completed, you have three values for insect larvae. Take out the mean for the distance traveled by that particular water meniscus in known time in 10 minutes. So you take the 10-minute constant. Then repeat the experiment with the same mass for germinating seeds and for single cell algae, repeating it with a fresh mass of carbon dioxide absorbent, using it fresh and using the same mass again. And all these organisms must be of the same mass as well. Repeat individual for 3 marks. This is what you will actually be telling here. So all of this should be repeated thrice. You get a mean there, and then this is all reliability as well. So then you can take out the rate of respiration as well. Rate is 1/time. So you can mention how the rate of respiration is calculated by 1/time. So that will give you get the rate of respiration. And the every time you will wait for 2 minutes. So safety, and for safety you will use gloves, because these algae and these seeds, they may have secretion that can stain, that can be irritant to the skin, not stain. They can be irritant to the skin. Plus the organism that you're using, they need to be, as I already mentioned, they need to be weighed on an electronic balance. So yeah, the electronic balance need to be mentioned as well. Apart from that, for example, for reliability, you will calculate the mean for all, already mentioned here. So if you're not using much of the electrical appliance there, but yes, electronic weighing balance is being used. So, and using water as well. So prevent yourself from minor electric shocks. So use electrical appliance with using dry hands. So precaution should be added together with the risk. So that pretty much covers everything you have to include in the answer. Further, they say the student calculate the rate of, yeah, and one more thing. If you're using germinating seed, the germinating seeds will start to photosynthesize as well. So you need to keep it in dark, or you need to just make sure that the this particular test tube is not having access to light, because germinating seeds will start to germinate if light is there. So that will start to add another variable, because photosynthesis is actually will end up using oxygen. So you need to prevent that. The student calculate, and you can have a control experiment as well. You can repeat it with control experiment by keeping glass beads of the same mass and repeating it and seeing that the water meniscus stays where it was. The student calculate the rate of respiration of oxygen units used per unit mass of the organism. Explain how the rate of respiration per unit mass. Per unit mass means for one, you will apply ratio method here. So what you can do, you can distance moved by the oxygen is noted from the capillary tube. We know the distance there. Now we need to convert the distance to volume. How you do that? But this particular capillary is like a cylinder. So the volume of the cylinder can be calculated using distance equal to into pi r square. You need to just remember you studied this formula in your math in your previous classes. This is not something that you discuss in bio, something that you already should be aware of. How rate of respiration can be calculated per unit mass? You will keep this volume of oxygen in cm cube here. The answer of this particular thing divided by time in seconds and mass in grams. The mass that you took for using the electronic balance, and the time that was calculated for the water meniscus to move. The student determined the respiratory quotient for each organism. To do so, the student need to measure the rate of CO2 production. Outline how the student could use the respirometer to find out the carbon dioxide productions. How that can be done. The carbon dioxide absorbent should be weighed initially before placing it, and eventually after the experiment should be reweighed. So difference in the mass recorded that shows CO2 produced, because it was the one which actually will add to the mass of the carbon dioxide absorbent. You divide the difference by the carbon dioxide mass by time, so that will give you the RQ value. Here, germinating seeds, insect larvae, single cell organism, their values are provided. RQ is already calculated for the one in 2 decimal places. For the other two, you will calculate in the 2 decimal places as well. You know the formula for calculating RQ. Just calculate the RQ for both. And if you are not familiar with how to calculate the value of RQ, just try to get the answer 0.72 by computing these. So if you get that, if you just cannot recall, so that's one thing that you can use. You can use the hint provided and use the same DP, the decimal places for your answer. With reference to RQ value in table 1.1, what conclusion can be drawn about the type of substrate respired by each of these organisms tested? So we know that we already have studied this, that if the RQ value is 1.0, it means that the carbohydrate is being respired by aerobic respiration. If it's 0.72, it means that lipids are being respired. If it's 0.80, it's protein being respired, or more appropriately, it's a combination of both the carbohydrate and the lipids. So that's you will be using. So here you just mention according to that, and you get 3 mark there. So again, you can see it's a 20-mark question. So as already mentioned initially as well in other videos, that in P5 one question is carrying more max as compared to the other question. So you need to have a good grasp of all the concept that you have studied. I hope this video was helpful for you. That's it from my side. Thank you so much.